All right, so introductory materials. And I'm just going to shoot through these first three because we've sort of already talked about it. Knowing, having a grasp that the sun is the main energy source for the planet is very, very important for us. All our energy, be that from plants or animals or what have you, it's all in the long run coming from the sun. Two, understanding the what do you call it, taxonomic tree of life, like what's a domain, what's a kingdom, what's a genus, what's a species. Understanding that a species is incredibly specific, whereas a kingdom is very broad. All right. Um, and on that sort of thread, if I were you, I would kind of be able to talk me through what a prokaryotic organism is versus a eukaryotic organism. So prokaryotic organisms are they lack membrane-bound organelles. They lack a true nucleus. They have what's called a nucleoid region where their DNA is located, and they tend to have like one big ring of DNA. Whereas a eukaryotic cell is very complicated. It's very complex. It's very fancy. They have membrane-bound organelles. They have like well, hang on. Let me not go off on a tangent too deeply here. But just a grasp of the difference between prokaryotes and eukaryotes. I would understand the difference between the archaeans and the bacteria. Where do you find them? Which ones are more prevalent? All right, archaea versus eukarya, or archaea versus bacteria. I would have a good grasp of the hierarchy of complexity. So, you being an organism made up of organ systems made up of organs made up of tissues to cells, cells to organelles to macromolecules to molecules, and those molecules being made up of individual atoms, which we're going to harp on endlessly as time progresses. Okay, like that is a main focus of the next lecture. Uh, all right, good enough. If I were you, I would have a good grasp of the scientific method. Just sort of, what comes after what, what are the bits and pieces in play here, and how they are employed. So, what's a hypothesis, and how does that lead to discovery, if you will? In scientific experimentation, I would have a decent grasp of, like, what experimental groups are versus control groups. What's a variable, basic knowledge of how science is done. Um, and I would have a grasp of like what a scientific theory is. Okay, theory in science is very important. It's not like saying, "Hey, I have a theory." No, 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 not the same thing. Scientific theory is like hardcore, broadly verified. All right, broadly verified. Let's see what else we have. All right, general characteristics of living things. Let me try to slow myself down here. General characteristics of living things. Um, living things are compartmentalized, as I described previously. Living things have the ability to change over time. Living things have the ability to reproduce and grow. Like, yeah, living things, characteristics, very important. Um, it's one of the reasons that we say, for instance, a virus is non-living, because they are acellular. If it's acellular, if it doesn't have a cell, then it can't be alive. All right, sure. Yeah, anything a something like asexual reproduction would be reproduction, or where an organism doesn't display sexual dimorphisms. Acellular means no cell, it doesn't have a cell. So if at all. So if you think about a cell, like in you've got this membrane, and inside of there's a nucleus, and then there's like rough ER and smooth ER and ribosomes and all that fun stuff, right? A virus is just a protein capsule, typically, with a strand of RNA inside of it, and that's it. Yeah, it's not a cell. It doesn't have a cell membrane. It doesn't have a cell wall. It has nothing internally but genetics. Yeah, pretty weird. We'll get there. Don't worry. Whole lecture on it. Give me time. Cell size. Okay, so you need to have a grasp that all cells have a given size. So, in other words, like your, I don't know, liver cells are pretty much the same size as a rat's liver cells, are pretty much the same size as a blue whale's liver cells. And the reason for that is that they all have a very defined, what we call surface area to volume ratio. Okay? Surface area to volume ratio. And that is to say that if cells get too big, they become, or let me rephrase, they lack efficiency. And when cells lack efficiency, they don't work terribly well. So if you can keep them small, you keep a lot of surface area compared to the volume, then they can diffuse products in and out very quickly and easily, and that vastly increases their efficiency. Yeah, that'll do. Okay. Alright, that's the first lecture, I think. Yeah, that's the first lecture. Let's go into lecture 2, that'd be chemistry, I believe. Yeah, go ahead. Oh yeah, you know it. That was only 7 bullet points on my little piece of paper. Literally, the last one says, cell size, efficiency. I know. It's a hard life. Alright, chemistry. Imagine those poor souls that aren't hearing this. Chemistry. So, let me see where to start. Let's just start with cells, or atoms, shall we? Start at the bottom, work our way up. You need to have a grasp of what atoms look like. If I give you a model of an atom, you got to tell me what the protons, neutrons, and electrons are. Okay, those subatomic particles. If I ask you what the charges are, you got to tell me what's positive, what's negative, what's neutral. If I ask you where the protons and neutrons are, you got to tell me it's in the nucleus. If I ask you where the electrons are, you got to tell me they're in energy clouds or shells along the outside. For that matter, you need to have a grasp of what would lead an atom to being reactive in terms of its valence shell. Does that sound familiar? All right. You got to know what's reactive in reference to its valence shell. So when would it be reactive versus when would it be non-reactive, like noble gases, for example. Let's see, what else do I have? Yeah, so noble gases have full valence shells from the get-go, all right? Whereas something like carbon has a need for more electrons, so it's capable of chemical bonding. Ergo is reactive. That's you're asking me a little something different. All right, give me a little time, and I'll think I might be able to explain that to you. All right, so we're going to talk about polarity in just a second. That may be where your brain's going. That doesn't have anything to do with noble gases, though. All right, so just give me a second. All right, where am I at? Oh, pH scale. So you need to have a grasp of the pH scale. 7 being neutral. As you go, oh man, people screw this up every time. I'll never forget sitting in an ecology class one time, and the professor just, you could see the look of complete sadness on his face as people consistently failed to understand that as the numbers go down, it becomes more acidic. Like lemon juice has a pH of 2, okay? Very acidic, very acidic. Whereas antacids have a pH that's much higher. That means they're basic, alkaline. Okay, so like a pH of 8 or 9 or 10 would be an alkaline, basic pH value. And you need to grasp that this scale is a log scale. Okay, it's logarithmic. What that means is that as one number changes to the next, it's a huge freaking change. Okay, a pH change from 8 to 5. No, let me try again. A pH change from 8 to 7, that's a 10-fold change because it's a log scale. Like the difference between 2 and 3, it's 10 times more acidic at 2 than it is at 3. Alright? So it's a log scale. Each number is very, very different from the next. Very different. Okay. So I've got acids and bases, pH scale. You might have a grasp of some of the human-related things along the pH scale. Wouldn't be a terrible idea. Just kind of look through it. Alright. Protons, neutrons, and electrons. You need to know about the atomic number versus the atomic mass. Okay? What does the mass tell you? What does the atomic number tell you? How can you interpret how many protons there are, for instance? What does that tell you about the number of electrons because they're balanced? Okay? So just having the ability to look at a model or look at a entry from the periodic table and kind of tell me what that means. It's not a terrible idea. Okay. Yep. Nope. Labs fill in the blank. We'll cross that bridge another day. But lecture is all either multiple choice, good old-fashioned standard multiple choice, or what I like to use a lot is the drop-down boxes that you've seen in other quizzes, right? So you click the little drop-down and it gives you a bunch of options, and you pick the one that's best. If you've taken my quizzes, you've seen these. So in other words, it's going to look just like the quizzes. Those quiz questions, by the way, pretty helpful for the test. Alright, polarity. Okay, this is, I think, where you were headed earlier. Having a grasp of polar covalent bonding versus non-polar covalent bonding. Remember, covalent means to share electrons, and polar covalency would be sharing them unequally. That's a result of what we call electronegativity. Okay? So these polar covalent bonds, they basically give a mild charge to certain areas of a molecule. Like, for instance, when we did water, you might recall that the oxygen side of water has a high electronegativity, so it attracts the electrons and it makes the oxygen side kind of negative. So then the hydrogen side of the water molecule becomes slightly positive, and that means that water molecules are adhesive and cohesive. That means that water molecules display hydrogen bonding. That means that water molecules give some unique characteristics, right? Didn't we harp on how water molecules have unique characteristics? Yes? I hope. Alright, that is from polar covalency. Okay, that's the side effect of polar covalency. Yeah, man, I would be good friends with the characteristics of water. Like, why is water so good at evaporative cooling? Why is water a wonderful insulator when it freezes? You know, when it freezes, it becomes less dense, and it floats on the surface and forms like a greenhouse for the water below it. So you can go up north and go to a frozen lake, drill a hole through it, reach down, and the water in there is not that cold. Alright? Because the ice on top is like a greenhouse system. It keeps the water below warm, and that sort of allows for life to survive in extremely cold environments. So one of the reasons that the Earth is in the shape that it's in right now, if you're not aware, the Earth has been frozen almost solid before. Alright, where am I at? Let's not go on that tangent either. Although I could. It's so much fun. My god. Alright, characteristics of water, water molecules, electronegativity, polarity. Yeah, okay. Pretty happy with that. That's chemistry. Okay, that's the whole chemistry lecture in a nutshell. On to organic molecules. Organic molecules. Alright, in terms of organic molecules, I would, let's see here. I would know the main functions and just general information, and I realize this is really an umbrella way of addressing this, but the main concepts behind carbohydrates, lipids, proteins, and nucleic acids. Those are the four main organic molecules that we discussed. Like, you need to be friends with these, man. Like, you need to know that carbs are sugars. In the carbs, there are monosaccharides, as single sugars, like fructose and glucose. You need to have a grasp that there are polysaccharides, many sugars bound together, like starch is a storage form of glucose in plants. You store glucose as glycogen in your liver and sometimes in your muscles. Plants have the ability to bind glucose in unique ways to form like cellulose. Right? Cellulose is what they use in their cell walls to give them strength. It's physically and chemically unavailable to us. Cellulose is glucose, but it's bound in a way that we can't utilize. Peptidoglycan is bound glucose in bacteria that allow them to have their cell walls. Oh man, chitin in insects. Like, you know, it goes on and on and on. Like these polysaccharides are just very, very important. Right? They are very important. And again, just so we're clear, starch in plants, glycogen in animals. Let's see, what else? Lipids. So in terms of lipids, I would be able to tell me about what a saturated fat is versus a non-saturated fat. Like, which one's of plant origin, which one's of animal origin, and kind of what does that mean for the molecules themselves. Tell me about steroids. You know, these are built from cholesterol molecules in the long run. They give rise to our secondary sexual characteristics. Steroids like vitamin D allow us to absorb calcium from our diet. Like, lipids are super important. Like, we have to have them. They really matter. Okay, they really matter. Protein. So, be able to tell me about amino acids. Like, you should be able to tell me about the building blocks to these, is what I'm saying. Tell me about amino acids. Tell me about how they peptide bond to one another. All right, what that really is polar covalency. But because this is protein, we call it peptide bonding, yeah? All right, these are referred to as being polypeptides. Like a big old honking protein's a polypeptide. It can take on multiple different shapes. Well, okay, I'm kind of running off on a tangent there. Let me see what else I've got. Proteins, polypeptides, polysaccharides, and sugars. Alright, on to DNA. So, in reference to DNA, I would know about its structure and function. Tell me about its double helix. Tell me about adenine, guanine, cytosine, and thymine, who they bind to. Tell me that DNA is a code for making proteins. Like, it's a cookbook, right? There's no mystery here. There's no mystery. like there's no magic. It's just a code for how to make protein. Alright, it's where your body stores the code for making proteins in your DNA. Understand that there are other nucleic acids out there, and we'll be talking more about those in just a second. But like RNA, that's nucleic acid in the long run. And ATP kind of looks like a nucleic acid. Not a terrible idea to understand that ATP looks like a nucleotide, right? Like, your DNA has a sugar phosphate backbone attached to these nitrogenous base pairs, and ATP does too, okay? Like, they look very similar. So some books will lump in ATP with the nucleic acids. Yeah, alright. That works. And then last but not least is the cells. Cells. And for those of you keeping track, we're 16 minutes in. Not bad. Not bad at all. Alright. My first overarching comment here is that I like to do a big matching block most of the time for this class, where I've got like A is DNA and B is RNA, and I've got like 10 questions. You gotta say if it's DNA or RNA. Like I'll say double-stranded, that's DNA. Contains uracil, that's RNA. Can leave the nucleus, that's RNA. Stores your genetic code for long periods of time, that's DNA. Like basically, who's who in terms of DNA versus RNA. I like that kind of thing. I think it's helpful. You need to have a good grasp of general cell theory. Like, what is a cell and what makes it a cell? General cell theory, not a bad idea to look it over. You can guarantee that I'll be asking about the differences between plant and animal cells. So, plant cells tend to be much bigger. Plant cells tend to have a cellulose-backed secondary cell wall. Okay? Plant cells have a big honkin' central vacuole. And you need to know about that central vacuole and what role it serves. Plants, this goes on. They have chloroplasts, right? Chloroplasts which allow them to do photosynthetic activity. Now, both plant and animal cells have mitochondria, but just plants have chloroplasts for photosynthesis. Okay? Or at least for our purposes at this particular moment, that's how this works. We're not going to get into some of the weird examples out there. Let's see, what else do I have for you? Yeah, plant and animal cells. You got to know the difference. Compare and contrast. Alright, inside of your cells, you need to know about the organelles, man. Tell me about the nucleus. Tell me about the rough endoplasmic reticulum and how it houses your ribosomes, right? Those ribosomes that make protein. Okay, they're the sites of protein synthesis. I would grasp that this is a eukaryotic cell we're talking about, whereas prokaryotic cells have that nucleoid region, right? The ring of DNA. Eukaryotic cells, by comparison, have multiple strand-like chromosomes in a nucleus. Okay, so ribosomes that make protein, like rough endoplasmic reticulum that houses those ribosomes, smooth endoplasmic reticulum which makes lipids in a lot of cases and does a lot of other things too, I might add. Golgi body that kind of shuffles things around to wherever they need to be. Yeah, man. All fair game. All fair game. Alright, you need to understand that mitochondria give rise to cellular respiration. Alright? And just kind of general structure of the mitochondria. Alright? Giving rise to cellular respiration. And understand that chloroplasts give rise to photosynthesis and general structures therein as well. Okay? Photosynthesis is to chloroplast as mitochondria is to cellular respiration. And then last but not least, team, I've got Cilia versus Flagella. Okay? What does Cilia look like? What do they do? How do they behave versus Flagella? Alright? They are, in fact, quite different. And I would say that's a pretty freaking sweet walkthrough of what you need to know. That's a good review. I think that's a good review of where we're headed with your... If you have watched my lectures and taken notes on them, if you have done my quizzes, which they're all up now, right? 1 through 4? Has anyone missed any of them? Good. Alright, if you've done the quizzes and you've looked over those again prior to the test, because it's not a bad idea to sort of see how I word my questions. If you have worked on the study guide and can now compare that to the conversation we just had, there's no reason at all that you shouldn't be pretty well set for this test. But don't sit there reading a page in black and white and think that just because you read it, you know it. That's not the case. You should probably consider, like Saturday, jumping on to the MyLab and Mastering and just seeing if you can work through some of their practice tests. Now, let me just be honest, their practice tests are probably terrible, right? They're built by people that don't know science. They look through the book and try to pull, like, random questions as best they can. They don't know what they're doing, so the questions are worded strangely. But if you can answer those, or at least you do them and then look at what the answers were supposed to be, it can't hurt. Alright, it can't hurt. So yeah, you should be all set. Anybody got questions? I got answers. Yeah, man. Sure. Trust me when I say it, people, when they're up to something, it's very obvious. Okay, it's very obvious. Like if, you got somebody comes running through the house or something, that's life. I understand that. If your phone rings, just don't sit there and talk on it for an eternity. If somebody comes in, don't have a conversation. And we're probably going to be in good shape. Alright? If you want to know what time it is, just don't worry about it. Just ignore if you want to, on your computer, I assume there's going to be a clock. Use that. Don't pick your cell phone up every 5 minutes and say you're trying to check the time. Yeah, it's not a big deal. You're not gonna break I'm a realist. All right, I'm a realist. I've been in the college world for a long time. I can tell when people are up to something, and they are not gonna benefit this semester. All right, I'm tired of it. I'm tired of it. Anyway, so yeah. So don't stress too much. Weird things happen, and I get that. I'm an easygoing guy. If you can't tell, maybe I sound like I'm not, but I swear to you, I am. I just had to censor myself. I was going to curse. Anyway, I promise I'm not a hard person to get along with. There we go. So yeah. Don't stress too much. As long as you're not clearly up to something. Like, it cracks me up. People wear, maybe I shouldn't say this. People wear glasses, and then they look down, and I can see them working on their phone, trying to look up something. And then the kicker to me is they email me afterwards and say, "Oh, I ran out of time. I don't think the time was fair." I'm like, well, if you weren't freaking trying to look stuff up, you'd have been fine. Anyway, yeah, one of those things. All right, anybody else? What you got? Yeah, it may be about to kick us off, so hustle. No, it's okay. Come on. Look, you're talking about cellular respiration versus photosynthesis, right? No. No, that's just my way of displaying to you the complexity of organic molecules. Remember, inorganic molecules are like 5, 6, 7 atoms, whereas organic molecules are hundreds of atoms in some cases. So I'm showing you just how crazy they are, by comparison. So the point there is to say, wow, organic molecules are amazing. All right? Sure. I believe that's going to be lab. Yep. That's correct. You want to do a review for lab? Is that what you're asking me? And we might can figure that out. It's another step up. Yeah. I understand. Yeah, we'll do this for the lab too. If you guys want it, if you guys are going to be here, I'll be here and work for you. So, but I probably won't make a recording because I want people to actually come in and do the work. All right, anybody else? Sure. 50 questions. I think I put 55 minutes on it. They will go very quickly. Like, I see no reason you shouldn't finish in 25 minutes, to be honest. Because it's going to go real fast. You just click, click, click. This should help a whole lot. I guarantee you, between the study guide and this conversation, I've mentioned everything on the test at least once. Yeah. Well, that's part of it. You know, there's a lot to learn. It's the whole study of life in a semester. Yeah, yeah. Yeah, I would leave that till last. All right. And by the way, it may kick us off in a second, and if it kicks us off and you still have questions, drop me an email, okay? Drop me an email. But yeah, I would leave. Hang on. I would leave my lab for last. I would worry about the study guide and this conversation and my lectures and my quizzes more than the my lab and mastery. Because I built your quizzes and I'm building your test. The my lab and mastering stuff, somebody else is doing those. That's the goal. Yeah, if you do the quizzes right, they should really help you study. And hint, hint, nudge, nudge, especially in lab. If you can answer the lab questions, hey man, you should be doing pretty good on the lab test. That is. All right, anybody else? Because they're about to kick us off in 2 minutes. All right, team. I will put this recording on the media gallery, and you should be able to access it later tonight. All right, later, guys. You have a great night, okay? I'm pulling for you. Do good. Do good. If you got any questions, email me. I'll check my email, like, Sunday mornings. If you got questions, I'll answer them. All right. Later, guys. Have a good one.